Our next speaker is Roberta Marino from University of Turin, and her talk is on integrative network-based approach, identifies gene communities in COPD.
Okay. Hi, everyone. I'm Roberta. I'm a PhD student from University of Turin in collaboration with GSK. So, okay. What about the biological framework in which we move? It is chronic obstructive pulmonary disease. It is the third leading cause of deaths globally, and it is characterized by having two alternating phases. They are the stable state from one side and the exacerbation state on the other side, also known as AECOPD. In a stable state, the patients are ill people. They have difficulty in breathing, but their symptoms are kept under control in some way. But there are also these other events which are called the exacerbations, where we have a sudden worsening of symptoms. We know very little about this event. We know that they are associated with presence in lungs of pathogens. But yes, we know very little, but we know also that exacerbations accelerate decline in lung function, and they increase also risk of mortality. So we got to better understand this kind of event. So we started analyzing data coming from this clinical epidemiological study, the IRIS, which has followed for 2 years patients with COPD. There were about 120 male and females. And we've analyzed blood gene expression during regular visits, so when they were stable, but also during the exacerbations. So we looked up in literature, and we found that there is an added value in integrating the gene expression with other data sets, gathering previous knowledge from experiments. So, yeah, what we've done basically is that of applying an integrative method, which worked like that, basically. First step, we've got a data set, whole data set gathering all the blood gene expression samples. We divide this data set into two groups, the one for the exacerbation group and the other one for the stable one. So we built two multinetworks. In these multinetworks, the nodes are the genes, and the layers, change based on the layers we look at. So for each multinetwork, the exacerbation multinetwork and the stable multinetwork or multiplex, we applied the InfoMap community detection algorithm for multinetwork. And then we found gene communities in one group and also in the other one. The other step, the next step was that of testing the enrichment of these gene communities with annotated biological functions. And then we compared these functions in these two groups, trying to see if there were some function specific for the exacerbation state. So that was a high-level description of the pipeline. So let's have a look at the multinetwork. So each multinetwork has 5 layers. The two networks which change in the two groups are the only one which change are the one based on the expression, the orange ones. And the ones based on the expression are these ones. First of all, there is a co-expression network which is built by the R package WGCNA. Then we also used, we built also another network which took expression as an input. Indeed, in this network, genes which have a close level of the expression in the same group of samples have a weight which is higher. Okay. Then we built also a transcription factor co-targeting network where the weight of the link is the number of transcription factors which regulate the genes. And also we built a microRNA co-targeting network, which is analogous to the previous one, but we looked at microRNA. And we also built a binary human protein interaction network. All these 3 layers are based on experimentally validated interactions. Okay, so in brief, here is the community detection algorithm we applied. It exploits the process of a random walk on a multi-network, where the probability to go from node J to node K, okay, it depends on the weight of the interaction of the link. And what is the link between this process of a random walk and the community detection in a network? Here is. Let's look at this network. In this network, the genes which have the same color have the same, belong to the same community. So we take a community, a partition, so a set of communities in our network, and there is this function, LM, which is a function of this partition. It is made up of 2 terms. The one describes the movements between the communities, and the one describes the movements within the communities. It is a sort of, entropy, an entropy expression to measure how the amount of information we need to describe our multi-network. So this algorithm aims at finding the minimal value for this function, LM. But it has a stochastic nature, this algorithm. So we applied it for 100 seeds, and we computed a consensus clustering. Okay. So after having the communities, we tested by hypergeometric test the enrichment of the genes in a community for annotated biological functions. What we got. So I've got to say that with the most classical analysis, the one which finds differentially expressed genes between exacerbation and stable state, there are no differentially expressed genes. But there are, according to gene set enrichment analysis, there are 3 pathways which are differentially expressed, the heme metabolism and something about interferon response, which are down-regulated in the exacerbation. But according to the multi-network results, there are some pathways which are specific for the exacerbation. Indeed, in this table, the rows are the biological functions. And here we've got 2 columns for the p-value, because in this column we've got the p-values for the exacerbation state, and here for the stable state. There are also 3 functions which are shared between the 2 groups. So after finding these functions which are specific for the exacerbation state, we asked ourselves about the link between the severity of the exacerbation and these functions which are specific for the exacerbation. And what we found in this graph, where the rows are the functions and the columns are the conditions, we found that the functions, so E2F target, estrogen response late, G2M checkpoint, cross signaling down, that were specific for the exacerbation, actually are specific for the moderate exacerbation, which are the majority in the pooled exacerbation group. And we also asked ourselves about the infection the link between the infection and the functions specific for the exacerbation event. And what we found was this thing. Look at this column. We've got an exacerbation with the bacteria in sporum, and we see that this function, the E2F target, the estrogen response, G2M checkpoint, the one specific for the exacerbation, actually are enriched when there is an infection. So summarizing, there are no differentially expressed genes if we contrast exacerbation and stable samples with COPD, but there are pathways which are differentially expressed, and there are also specific functions which are enriched according to the multi-network analysis. These functions are specific for moderate exacerbation, but also for exacerbation with bacteria, with an infection in lungs. This network approach has been used also to answer other questions for the IRIS dataset. There are some examples here, and also other different GitSet collections have been used to do the enrichment test. So future developments, it would be interesting to look at a dataset with healthy subjects. Indeed, in the IRIS dataset only contains people who are ill, who have COPD. So we don't have a real control. And it would be also, yeah, I'm also analyzing, I'm doing the same analysis, but with multi-networks, so with only the expression layers to see what we gain using also the other networks. And I would like also, I'm going also to apply the Laura Cantini pipelines I cited before, because she also analyzed, used the multi-network approach to contrast tumor and healthy subjects, healthy samples, to see if there is an agreement in results. And yeah, I would like to thank you all for your attention, and also all these people who helped me a lot, in particular Alessandro Muzzi, my mentor, and Michele Caselle, my mentor for the University of Turin. Thank you.
We have plenty of time for questions. Yes, please.
Thank you very much for your talk. I was wondering that which method did you use for deriving differentially expressed genes?
Okay. The Lima has been used, but it has not been used by me, but Joseph El Alamat, but used Lima, so linear regression. But I don't.
And what was the threshold for significance of these differentially expressed genes for p-value, I mean?
0.05.
I see. Because I was very wondering that why you didn't find any differentially expressed gene usually.
Yes, I saw in the literature that it was not so difficult to find in COPD because, for example, when they contrasted in publication by, I remember Hirsch, where they contrasted samples with high frequency of exacerbation and low frequency of exacerbation, there were no differentially expressed genes, but there were pathways which were differentially expressed.
Okay, thanks.
Are there any other questions?
I don't know if.
Can you please turn off your mic if you're done with questions? Thank you. Thank you. Can the next